All organisms require a mechanism of DNA replication. For simple organisms, conservative replication is used. This is the process of producing a copy of a double helix. However, DNA replication is more commonly examined under the semi-conservative model, used in complex organisms including humans. The first step is strand separation. An enzyme called helicase will unzip the two DNA strands in the 3' to 5' direction. This separates the strands by breaking hydrogen bonds between the nitrogenous base pairs. The double helix begins to unwind, which causes a supercoil to occur at both ends of the replication bubble. The enzyme topoisomerase will break a single phosphodiester bond which releases the coil. As a bond has been broken, single-stranded binding proteins must connect the strand to prevent it from floating away. This process will occur concurrently throughout the DNA strand for maximum efficiency. Now that we have two separate strands, the DNA can begin strand synthesis. In order to do this, we need another enzyme called DNA polymerase III. This reads the leading strand from 3' to 5', and creates a complementary daughter strand from 5' to 3'. Since helicase moves in the same direction as polymerase III, this process can create a fully duplicated strand. While this is relatively straightforward, things become more complicated for lagging strand synthesis, as helicase moves in the opposite direction of polymerase III. This means that the section of DNA downstream from polymerase III will not get copied, as polymerase III can only read from 3 to 5'. In order to solve this problem, the enzyme RNA' attaches pieces of RNA nucleotides downstream, which provides a primer for polymerase III to attach, which then reads and synthesizes a new strand. However, as the strand is now a mix of RNA and DNA along with missing phosphester bonds, called Okazaki fragments, it must be remedied. DNA polymerase I will replace the RNA nucleotides with DNA nucleotides, and DNA ligase will place the missing phosphester bonds between the fragments. Polymerase III will proofread the new strand and go backwards if any mistakes were made. It will also look for incorrect base pairing. If there are any errors, it will be replaced with the correct nucleotide. Replication is now complete, and the new DNA strand is ready to be used. This process is incredibly important, as every time a cell divides through mitosis, DNA must be replicated. Without it, our cells could not be replaced when damage occurs to our body, such as cuts to the skin,